Hi friends, today we will be talking about one of the central what you call as a catabolic cyclical pathway involved in the oxidation of acetyl-CoA. A complete oxidation of acetyl-CoA to form carbon dioxide and formation of energy, rather storage of this energy in the form of the reduced electron carriers like your NADH, FADH2, which can be later on oxidized in the electron transport chain for synthesis of ATP. So we basically, when we divide the what you call as reaction sequences in the complete oxidation of the glucose, the first step which we see is glycolysis. The second intermediate, which actually converts your pyruvate into acetyl-CoA as a link reaction, and the second major what you call as the catabolic pathway which is involved in the oxidation of this acetyl-CoA is this citric acid cycle, the citric acid cycle or the TCA cycle, the tricarboxylic acid cycle. Most of the intermediates which are formed here, they contain 3 carboxylic acid groups, as you can see here, this one, this one, this one. So it has 3 carboxylic acid groups, and therefore this particular cyclical pathway is called as the tricarboxylic acid cycle. We also sometimes refer to as this as the Krebs cycle because it was discovered by Sir Hans Krebs. We call this particular pathway as the Krebs cycle. So in biology, as you know, that most of the times it is the names which are very confusing. So let's have a revision of these this nomenclature. So the pathway, whether it is the Krebs cycle or the TCA cycle or the citric acid cycle, it means the same. Okay, so as I told, talking about this citric acid cycle comprising of the 7 cyclical steps, which will start with oxaloacetate and it will end with oxaloacetate. So total in all we have 8 steps excluding the first step of condensation. So this particular entire pathway will start from oxaloacetate and it will end back to oxaloacetate. So talking about this pathway, if you look at the first step, catalyzed by the enzyme citrate synthase. Now this particular citrate synthase catalyzes the very first step of what is called as your condensation. So there is a condensation of a 4 carbon oxaloacetate. Oh, here it is. So you have this 4 carbon oxaloacetate condensed with a 2 carbon acetyl-CoA, which we have got from after glycolysis and after the link reaction, then we have got this acetyl-CoA. So this acetyl-CoA will condense with oxaloacetate. The CoH group will be removed and you have the formation of the first intermediate, that is your citrate. Therefore this particular pathway is called as the citric acid cycle, because the first intermediate which is formed in this cyclical sequence is the citrate. So remember, this is called as citric acid cycle Q because the first product which is formed is the citrate. This is called as the Krebs cycle. Why? Because it was discovered by Sir Hans Krebs. And this is called as the TCA because most of the intermediates, they have 3 carboxylic groups, and therefore the tricarboxylic acid cycle. The citrate itself has 3 carboxylic group, as you can see. This then undergoes the successive dehydration and hydration. So removal of water and addition of water, making some molecular rearrangements. So there is a removal of water here to form a temporary intermediate, that is cis-aconitate, and then this cis-aconitate is again hydrated to form isocitrate. So your second step, we divide into 2A and 2B. They occur very quickly, catalyzed by the same enzyme, that is your aconitase. So aconitase enzyme, it will convert your citrate into isocitrate via the intermediate formation of cis-aconitate. So dehydration followed by hydration. So remove water, add water, and you form what is called as your isocitrate. So this was 4 carbon atoms, right? This was 2 carbon atoms. So this you have a total of 6 carbon atoms. So here again you have isocitrate which is of 6 carbon atoms. The next particular reaction now is it's time to harness the energy. So the next step is that of oxidative decarboxylation. So after these 2A and 2B steps, the next step of oxidative decarboxylation, as the name suggests, there is both oxidation as well as decarboxylation. So removal of hydrogen in this particular terms and removal of carbon dioxide. So you have isocitrate being oxidatively decarboxylated. So after oxidation, right, there is a removal of these electron pair, removal of this hydrogen which will be accepted by your NAD. and NAD, it will be converted to your NADH and H plus. So NADH and H plus formation takes place. So your first NADH is formed. This is the storehouse of energy, or this is the reducing equivalent which you have, which you can later on use for multiple purposes. Here we generally use this for generation of energy in the form of ATP. In our next lectures probably we will see that how one NADH molecule can give around 2.5 molecules of your ATP later on. So isocitrate dehydrogenase will remove a carbon dioxide, and isocitrate will be oxidized and decarboxylated to form alpha ketoglutarate. So this isocitrate was of 6 carbon atoms. 1 carbon atom is gone. So this alpha ketoglutarate is made of 5 carbon atoms. So you have the intermediate formation of what is called as alpha ketoglutarate. Now this alpha ketoglutarate is acted upon by the next set of enzyme called as alpha ketoglutarate dehydrogenase. Now these are actually enzyme complexes, though it is appearing as a single enzyme. This is not a single enzyme, you have multiple enzymes working together. So it is the alpha ketoglutarate dehydrogenase enzyme complex. So this alpha ketoglutarate dehydrogenase enzyme, it will again catalyze the oxidative decarboxylation. So remember, there are 2 steps of oxidative decarboxylation. First, when your isocitrate gets converted to alpha ketoglutarate. Second, when your alpha ketoglutarate gets converted to your succinyl CoA. So your alpha ketoglutarate now got converted to succinyl CoA. There was a loss of carbon dioxide and there was oxidation. So electrons were removed, hydrogen atoms were removed, they were again accepted by your NAD. So you have the formation of the next molecule of your NADH and H plus. So there is a oxidation, formation of NADH, and there is a decarboxylation, removal of carbon dioxide. So this is what happens here. So this is catalyzed by your alpha ketoglutarate dehydrogenase. So your 5 carbon atom alpha ketoglutarate now gets converted to succinyl CoA, which is a 4 carbon atom intermediate. So your succinyl CoA, here it is, succinyl CoA now is acted upon by succinyl CoA synthetase. Now as the name suggests, the synthetase, it results in the formation of what is called as your GTP. Say a cousin brother of ATP, real cousin. So either you can say like GDP gets converted to GTP, or your say ADP gets converted to ATP, the step which we refer to as substrate level phosphorylation. We all have studied, right? Whenever the electrons are removed and they are what you call as temporarily harnessed here as NADH and H plus or FeDH2 and later on oxidized for liberation of energy in the form of ATP, we call this phenomenon as oxidative phosphorylation. Here, while one particular substrate is being converted to the other product, there is formation of a high energy compound like GTP or ATP. This is called as substrate level phosphorylation. You've encountered this substrate level phosphorylation in glycolysis also, where your PEP got converted to pyruvate, there was formation of ATP. So similar kind of thing it happens here. So here your 4 carbon atoms are here, succinyl CoA got converted to succinate, the CoAsh group which you have added, that will leave the group. The energy which is stored is harnessed in the form of your GTP. So GDP gets converted to your GTP, and then this succinate is acted upon by succinate dehydrogenase. So the next step is what is of dehydrogenation. Many of the times we are confused with dehydration and dehydrogenation. Remember, whenever we are talking about hydration, we are talking about water. Like we talked about here, it was dehydration. So it was removal of water molecule. Here I am talking about dehydrogenation. It is removal of hydrogen atoms. It is an oxidation reaction what we talk about. Succinate dehydrogenase has FAD as the cofactor which accepts these electrons. So these electrons or hydrogens, they are accepted by FAD and FAD gets converted to FADH2. So there is a net formation of what is called as your FADH2. So fumarate is formed, succinate is oxidized, I can say, to form what is called as your fumarate. So here you have the formation of what is called as a fumarate, a 4 carbon intermediate. So this fumarate now is then hydrated. So there is a reaction of hydration. So water is added and there is formation of what is called as your malate, catalyzed by the enzyme fumarase. So this fumarase adds the water across the double bond and there is formation of what is called as your malate. Now we have come at the step where we again regenerate from where we started. You again oxidize this malate to oxaloacetate. So during oxidation, there will be loss of electrons, loss of hydrogen. So again, you have acceptor as your NAD. So again, there is formation of NADH and H+. So malate dehydrogenase oxidizes your malate, this particular one, to your oxaloacetate. So there is a oxidation. So here there is an introduction of this double bond and there is formation of this NADH and H+, and the cycle is completed. If you see, 2 carbon atoms, they entered here in the form of acetyl-CoA, they are removed or they are oxidized in the form of carbon dioxide. So there is a complete oxidation now. So for complete oxidation of that glucose which had entered, which you oxidized in stepwise manner to pyruvate, then you converted it to acetyl-CoA, it is now completely oxidized and liberated as what is called as your CO2. So this is how the citric acid cycle it operates. So let us go one by one. So in the first step, oxaloacetate combines with acetyl-CoA to form citrate. Citrate via cis-aconitate formation gets converted to isocitrate. Isocitrate is oxidatively decarboxylated to form alpha ketoglutarate. Alpha ketoglutarate is again oxidatively decarboxylated to form succinyl-CoA. Succinyl-CoA, after substrate level phosphorylation removal of CoH group, gets converted to succinate. Succinate oxidized to form fumarate. Fumarate hydrated to form malate. Malate again oxidized to form oxaloacetate. So basically, if you sum up, what has happened in the citric acid cycle is your acetyl-CoA molecule has been completely oxidized to CO2, number one. What other things have happened? We have generated 3 molecules of your NADH. So one pass through the citric acid cycle, it will form 3 molecules of your NADH. One pass of the citric acid cycle will form 1 molecule of your FADH2. One pass of the citric acid cycle will form 1 molecule of your GTP. So to summarize, as I said, we have formation of 3 molecules of your NADH, 1 molecule of your FADH2 and 1 molecule of your GTP equivalent to your ATP which is being formed. So this is what happens in a typical citric acid cycle. Just let me know that what will happen to this particular products which are formed when you start with 1 molecule of glucose. To just give you a primary hint, you start with 1 molecule of glucose, you end up with 2 molecules of pyruvate, and these 2 molecules of pyruvate will be converted to 2 molecules of your acetyl-CoA. So just tell me how many molecules of your NADH, FADH2 and GTP will be formed if this citric acid is operating to oxidize 1 molecule of your glucose. This is what you have to tell me. Okay. One more thing to mention here, that though this we consider as a catabolic pathway, many of the intermediates here, they are used for synthesis of various biosynthetic precursors. So your alpha ketoglutarate may be removed, your succinate may be removed. So they can be removed and they can be used for biosynthesis. You can use them for synthesis of amino acid. You can use them for synthesis of purines, for synthesis of pyrimidines or many other such molecules. So once these particular intermediate is removed, some anaplerotic reactions, they replace them. And again, if there are any 4 or 5 carbon intermediates which are formed during the oxidation of any other amino acid or any other fatty acid, they can enter into this pathway at any particular moment and this pathway will continue. So citric acid cycle summed up, there are many more things. Stay tuned with me, Professor Girish Kukreja, for more in microbiology and biochemistry. And yes, don't forget to tell me what happens with 1 glucose, how many NADH, how many FADH2 and how many GTP. Thank you.